All right, so welcome to part two of genetics. Now, this is where things get fun. While Mendel had done a lot of really cool research on genetics where he was able to discover the dominant and recessive factors, and now we call those alleles, and go through and show how inheritance occurs within continual generations. When we start looking at genetics outside of pea plants, like in humans, it gets to be much more complicated. So, pea plants, remember, were very simple. They were great model organisms, great test organisms in that sense. But if we start looking at humans, we actually see that while some of the traits in humans can be the dominant recessive inheritance, we also can't do easy things like test crosses, right? If you try to test cross a human, there's a lot of complications with that. Ethical, money, time, it takes 9 months to make a baby, and then you're forcing people to make babies with whoever you want them to. Obviously, it becomes a little more difficult. So if we want to look at pattern of inheritance within families, within human populations, we can still apply Mendel's laws, but we're going to look at them in a slightly different way. We're going to start by talking about pedigrees. When we look at human characteristics, we often look at things called pedigrees, which are these kind of charts, these like family tree charts that you might see. Now, when we read a pedigree, there are a couple of ways to go about it, okay? First, where is my little pen? Oh, I lost my pen. That's okay. So first, when we talk about pedigrees, one of the things we need to note is that males are squares and females are denoted as circles, okay? So in this instance, in this pedigree I have two males and three females, okay? This is a family, and we assume familial relationship here. We assume inheritance because this male and this female are showing that they have a union. Usually it's a marriage, but it could be just a union of some sort. And these two individuals report having these three children. So the first two individuals are a male and a female that report having a son and two daughters. Okay. When we look, then we can also see that if an individual is blank or not shaded in, it's just a square or a circle without any sort of coloring, then that individual does not have whatever trait we are tracking, whether that trait be dominant or recessive. Instead, they are going to just be, they just don't have that trait. Okay. Individuals that are shaded in do have a trait. And so in this instance, we see that these two individuals had a female offspring who had the trait that we are looking for. Okay. And again, it doesn't have to be a dominant or recessive trait, particularly, it could just be a trait. Now, in this instance, we have to make some assumptions, okay? And this becomes really interesting when you start looking back at like royal family lineages. Pedigrees are going to be, we have to assume everyone's telling the truth, okay? We have to assume that Mary Sue here did not have a baby with somebody else, but actually had all three of her children with this individual, say, Bobby, okay? We have to assume that this is true. And in general cases, yes, okay? However, if you look at like the royal family tree, you're going to find some instances where that wasn't the case. But if I was the queen, I would not want to admit that my son, my potential heir to the kingdom, was not the king's son, right? And so I'm going to say that it's his, even if it's not. So when we're looking at these traits, okay, another thing that we do when reading pedigrees is we actually denote individual generations by Roman numerals. So generation 1 is going to be our first group of individuals, and then 2, and then 3, and then 4, okay? And we could then use numbers, the numbers that you're used to denote the individuals on the tree. So we could say that individual 1 is this female who happens to be autosomal recessive for the trait that we're looking at. Okay, individual 1, 2 is the male, and we don't know what his second allele is, but we know that he has the dominant phenotype, and so he has to have the dominant allele, at least in one of his two alleles. Okay, so this particular family tree is illustrating an autosomal recessive disorder. That means that this is going to be on the chromosomes 1 through 22, so chromosome pairs 1 through 22, and it happens to be recessive. So individuals who have it, we know their genotype is going to be homozygous recessive because they have the recessive phenotype. And remember, we can tell recessive genotypes from recessive phenotypes. But other individuals, we may or may not know if they are dominant or heterozygous, okay? So we have an individual here who had offspring with this male, and she happened to be homozygous recessive, okay? They had two daughters. Now, we know that their daughters are going to potentially be heterozygous, okay? Um, Now, it's weird because we have it filled in here, but that's because we usually work backwards on pedigrees. But let's just say that they only had 2 children. In that case, that is not enough offspring to determine what this gentleman's second allele was. Because if they had 6 children and all of their children were heterozygous, then there's a good chance that they have that he's homozygous dominant. But that's not always the case. Okay, I had a student a couple semesters ago who had 7 children. Her first 6 children had brown eyes, and she and her husband had brown eyes. And so she was assuming that she was homozygous dominant and he was homozygous dominant, right? Or one of them was homozygous dominant and the other one could have been heterozygous because blue eyes is recessive to brown eyes. But their 7th child had blue eyes, which means that they happen to actually be heterozygous. But remember, every time that we go through that Punnett square, it's the same sort of frequency, right? The same chance of having those alleles inherited. And so even though they had 6 children with brown eyes, we couldn't determine for sure that what their alleles were until they had their 7th child. At that point, we know that they have to be heterozygous because they are carrying the allele for that recessive trait. Okay. And that's what ends up happening here. So if I go and I look and I'm saying, okay, look, this second generation, nobody has the trait that I'm looking for. The third generation, nobody has the trait that I'm looking for. Okay, but this fourth generation, I have 2 individuals with the recessive allele. And now again, this could be something simple like blue eyes. It could be something more problematic like cystic fibrosis. But in any case, we see that these last 2 individuals, individual 4-1 is a female that's affected, and individual 4-2 is a male that's affected. What you might have noticed is that individual 3-1 and 3-2 are cousins. I know, but this is not as abnormal as you think, and it's happened a lot throughout history. So, like, even Charles Darwin married his cousins. Like, don't panic, okay? But we do know then that these 2 individuals are heterozygous because their children inherited recessive alleles. And as such, we have to assume that their mother is heterozygous on both sides because their mom was heterozygous, okay? In any case, or homozygous recessive, the mom was. In any case, this is going to be an autosomal recessive disorder because we skipped generations, okay? We've skipped generations. It's not every generation that's affected. And the children that are affected are male or female. It doesn't matter which sex it is. So it's autosomal recessive disorder. Okay. There are a couple very common autosomal recessive disorders, or fairly common ones that we might be familiar with. The first being cystic fibrosis. This is a very common illness. It actually results in these thick layers of mucus, whether in the respiratory system or the intestinal system. And it actually stops your cells from being able to do some of the ion exchanges that they need to do. And it can be really problematic. One of my good friends in high school had CF, and I watched that man eat 16,000 calories in one sitting because his CF particularly affected his digestive tract, and he actually didn't absorb nutrients from the majority of the food. So even though he was eating 16,000 calories, he was incredibly thin because he's actually not breaking down and digesting that food effectively because the mucus lining his intestines was blocking his ability to absorb those nutrients. Okay. He was one of 3 children. His sister also had CF. And so again, it's just that reminder that every reproductive event has the equal chance of being recessive if it is a recessive allele that can be inherited. Okay. In addition, we see things like autosomal recessive and sickle cell disease. And I talked about this a little bit in chapter uh, 3 when we were talking about our biomolecules, and I talked about how sickle cell anemia is an inherited trait that is a recessive disorder that changes a single amino acid in the protein sequence and results in hemoglobins that do not that bend or fold or are sticky, and they end up causing clots and can cause a lot of pain and inability to do exercise and things because they can't move oxygen around their bodies as effectively. Individuals with this are going to be autosomal recessive. And one of the things that's interesting about this particular disorder is it's very common in people from West Africa who are relatively, the population has been exposed for a long period of time to high rates of malaria infection. And one of the things that we think might be happening here, the hypothesis, is something called a heterozygote advantage. And basically, individuals who are heterozygous are able to potentially resist malaria. And so we see more individuals who are heterozygous in that population because individuals who are homozygous dominant might get malaria and die from the malaria because they can't resist it. And malaria is still one of the most deadly diseases to humans. Okay. And individuals who are homozygous recessive may pass from clots, especially if you get clots in your lungs or heart or brain, obviously not a good thing, as a result of sickle cell disease. But individuals who are heterozygous are able to adapt and survive because they don't have the sickle cell disease and they are less affected by malaria. And so this trait is maintained in that population simply because we have a lot more heterozygous individuals in that population. In any case, there's a lot of really cool treatments that are going into sickle cell disease, especially some really interesting ways of gene therapy to try to replace. We know what that error is, we know where it is. And so there's some really cool gene therapy to try to reverse this in individuals who have this because it can be a very debilitating disease. Okay. When we have autosomal dominant disorders, we can tell from a pedigree because every generation is going to have individuals who are affected. And if you inherit the allele, whether you are homozygous dominant or heterozygous, you will have the trait. Okay. The child could be unaffected if the parents are affected, because if the parents are heterozygous, they can have children who are homozygous recessive. Okay. In this case, we see that it's both males and females equally affected, and so this is going to be autosomal. It's not on the sex chromosomes. And we see that it is in every generation of individuals who are reproducing from this first generation, these first two individuals. Now, again, not every autosomal dominant trait is going to be a disorder or a genetic disease. One of my personal favorites is polydactyly. So polydactyly is having extra digits, and having 5 digits might be the wild type in humans. That's the norm, the more common, but it is actually a recessive trait. Having extra digits is a dominant allele. And I have a fantastic example of this. In my neighborhood, there are 2 cats that live down the road. I like to go visit them on my walks, and their names are Toes and Fluffy. Okay, so I'm assuming you can picture what those cats look like in your head. They're both orange, they're very sweet, they're from the same litter. Okay, Toes has extra toes, she has extra digits. Okay, her brother Fluffy does not have extra digits. So that tells us that their mom was heterozygous for that trait, and because we know their mom had extra digits, Toes inherited the dominant allele and Fluffy did not. Fluffy inherited 2 recessive alleles. However, he is the fluffiest cat I've ever seen, and they're both great, but they're a huge example, a great example of how autosomal dominant traits can be inherited in some individuals and not others, even from cats from the same litter. Okay? So, let's talk about some of the disorders, or especially the disorder that we do know about is Huntington disease. Huntington disease is just absolutely devastating. It's a progressive degeneration of the neurons in the brain, which can absolutely lead to huge problems. The sad thing with Huntington disease, it is dominant, and so if an individual has it, they will potentially pass it to their children, actually have a higher than normal rate to pass it to their children with that dominant allele. The problem is Huntington disease can spontaneously arise in human lineages and is not necessarily identified early on. Individuals who are the first generations to have Huntington's disease might not even know that they have it until they're in their late 50s, early 60s, maybe even a little bit later. Most people by their 50s or 60s have children and probably grandchildren, and so they've already passed their genes on, and their children have potentially already passed their genes on before anybody even knows that Huntington's is running in the family. Unfortunately, it does become progressively worse with each generation. This is another one that is targeted for gene therapies because we do know where that trait is coming from, and there's been some potential there. So, we'll see what happens. Most people, if they have a risk or they're concerned about it, you can absolutely test for the presence of this gene, as well as many other genetic disorders that may run in your family. Okay, I personally have a hypermobility disorder, and so like I know that that is something in my family lineage. My brothers have then had their children checked just in case, so that if they have a hypermobility disorder, they can be aware of it early on. Okay. All right, make sure you can check your understanding for these traits, especially for reading pedigrees within human characteristics. Once you can answer these questions, go on to the next part, which is the weird things with Mendel's laws. Okay. All right, so Mendel's laws do cover the simple or basic dominant recessive inheritance, right? Where if you have a dominant allele, it covers the recessive allele, and that's very, very straightforward. However, once again, that is not always going to be the case, and that's definitely not the case with humans, and it's definitely not the case with a lot of organisms. So let's talk about a couple of ways that what we know today has kind of gone beyond what Mendel was studying and understood in the 1860s. Okay, that was a long time ago. So, one of the ways that we see this is something called incomplete dominance. This is also known as blending, or like, I think incomplete dominance makes sense. So, incomplete dominance is when the individuals who are heterozygote have a different phenotype than the individuals who are homozygous dominant and homozygous recessive. And this is because the dominant allele doesn't fully mask what is considered the recessive allele. So I have wavy hair. I have a couple pieces that like ringlet curl and it does its own thing. That is a trait that is incomplete dominance. Okay, if you have wavy hair, it's between curly and straight hair. My dad has very, very curly hair. My mom has very straight hair. I got the in between, the mixing of those two alleles. Okay, a common way we see this is absolutely with flowers. Flowers do this a lot, where if I have a red flower and a white flower and I breed them together, their offspring are pink. Well, pink, we know, is, if you know your color theory and your color wheel, pink is going to be made when red and white are mixed. And so it makes sense then that my individuals who are purebred and red will always have red flowers. My purebred white individuals will always have white flowers. And so I would think that they are dominant and recessive alleles. But when I actually cross red and white, my first generation will be pink. And you're like, okay, cool, right? That is an example then of incomplete dominance. And if I allow those pink individuals to then cross, my next generation will show a 1 to 2 to 1 phenotypic ratio. And that ratio is going to be a little bit different than what we see in the 3 to 1 ratio, but the genotypic ratio is still 1 to 2 to 1, but my phenotypic ratio is also 1 to 2 to 1. Okay. Other examples of this include like roan horses, or if you have a brown and a white dog and the puppy is tan, that sort of thing. Now, when we look at incomplete dominance, we have something that's kind of the opposite, where we would call that codominance. Codominance is also known as a multiple allele trait. And this is when more than one dominant allele exists in that population, and both can be equally expressed if they are inherited. The classic example of this is the ABO blood typing in humans. I like this genetics section because I get to talk about how I'm an example of like all of these things. So I have an AB blood type. Okay, I am AB positive, which means that I have the A antigen and the B antigen on my red blood cells, and that means that both of those are equally expressed in my body. It also means that I'm the universal receiver, so I can get O blood, A blood, B blood, AB blood, doesn't really matter, okay? Any of that I am not going to have an issue with. My husband, on the other hand, is an O blood type. So my husband has the O blood type, which is actually the recessive, and basically it means you have neither A nor B. You're missing the O, okay? And so in that case, he is homozygous recessive, and I have a dominant A and a dominant B. Isn't that cool? My brothers are both A blood type, and my stepson is O blood type. And so while my stepson looks like me, he's very blonde, he's very blue-eyed, he looks like me, There's no way he could be my child, because if my husband and I were to have a child, our child would be either A or B a blood type, because they would inherit the dominant allele from me, either the dominant A or the dominant B, and the recessive allele from my husband because he's O and he can only donate the recessive allele. It's kind of cool, right? So you could kind of look back and think maybe about your own your own family's blood typing. I had a student one semester who had 4 children, and they were all O blood type, and it turns out he and his wife were both O blood type, and so their whole family was recessive for the blood typing, and it's kind of a cool thing there. Okay, so codominance. This is also sometimes seen in flowers, where if I have a red and a white flower that cross, I don't get pink offspring, but I do get red and white striped or red and white splotched offspring. That would also be an example of codominance. So incomplete dominance and codominance are kind of opposites of each other. One, we have a blending, one, they're equally expressed if you inherit the 2 dominant alleles. And then we have 2 more that I want you to know as far as these kind of weird ones. So there is polygenic inheritance. Polygenic inheritance is honestly the most common in humans, and actually most of our physical characteristics, our hair color, eye color, skin color, height, weight, are examples of polygenic inheritance. Remember that poly means many, genic is genes, and so this literally means many genes are influencing one trait. And so each of these are going to have an effect as an overall, and it results in a continuous bell curve when we look at a population. But what it means is that you can have someone, there are actually 3 sets of genes, so 6 alleles that can determine human height. And so sometimes you can have those families where one individual is really, really tall and one individual is short, even though they're siblings. And it's just because which of those alleles they inherited and how they are influencing that characteristic. Again, same thing with hair color, eye color, skin color. We are going to see a lot of these are polygenic traits. Some of the things that we know as common disorders could also potentially be polygenic, such as allergies, cancers, diabetes, schizophrenia potentially. So there are a lot of ways, like it makes sense though, right? We have 23 chromosomes, pairs of chromosomes. There's a lot of genes there. It would be really odd if we just had one gene that was a set of height, okay? So that's polygenic. And then the opposite of polygenic, where instead of many genes controlling one factor, are single genes having more than one effect. And so this is something that is called pleiotropy. And it's lots of genes having more than one effect. It's kind of a cascade issue. So sickle cell anemia, again, is an example of this, because if you have sickle cell anemia, you are going to have like exercise intolerance, higher chances of clots. You are going to potentially have bruising and other issues that become a problem. And that's all because of one wrong gene, right? One mutated gene, one set of alleles that is recessive. Another example are hypermobility disorders. So Marfan syndrome is an extreme hypermobility disorder, but most hypermobility disorders are connective tissue defects, and it can result in skeletal issues. So I had mentioned I have a hypermobility disorder. I have something called Ehlers-Danlos, and it results for me in having very loose joints, bruising very easily, like incredibly easily. Luckily, I am not showing any signs and symptoms of heart and blood vessels, eyes, or lung issues, but my skin is stretchy, and I'm actually having some issues with arthritis because I have hyperextended, dislocated, subluxed my joints so many times that I'm actually having some issues with that. And that's because of these traits that are kind of cascaded from a single inherited allele. Okay. And again, it's one set of alleles rather than many sets of alleles. This is one set of alleles that will have a cascade effect. But connective tissues like hold your whole body together. So it makes sense that if you have a disorder in a connective tissue, you're going to see other things happening. Same thing with sickle cell anemia. Those are your red blood cells. They do so much work in your body. We're going to see a cascade effect from that. Okay, so those are the 4 different ways that we see things going beyond Mendel's dominant and recessive inheritance. We're also going to talk about sex-linked inheritance in humans because it's interesting. Okay, so remember that when we're talking about someone who's biologically female, they are XX, and again, range of things, intersex, right? We're keeping this basic for Bio 110. In this case, eggs are being produced by an individual who is XX, and as such, the female is donating the X chromosomes to all of her offspring, right? Because all she can donate is an X. Males are XY, and so the sperm can either contain an X or a Y, and so the sperm is actually what determines if you are producing a female child or a male child. The females are going to have, again, we're 23 chromosome pairs, so 22 autosomes plus the sex chromosomes. So 44 autosomes plus XX, 44 autosomes plus XY, remembering that XY are not homologous in that case. And Y, again, it's just like 200 genes, and it really determines maleness. X is super important. There are 2,000 genes, and they are really important for the physical development of an individual. So you need to have an X chromosome. As such, all individuals are going to inherit an X from their female, from their mother. The X chromosome can carry a huge array of genes. And again, the Y is not going to have those associated genes. So one of the really common sex-linked disorders that we talk about is things like color blindness. And males are more often likely to be colorblind than females, and that's because females inherit two X's. When you inherit two copies of an allele, you have a higher chance of being heterozygous. Even if you get that recessive allele, you have a higher chance of having a second allele that is dominant. Males inherit one X chromosome, which means that if they inherit a recessive X, that is what they've got. They do not have a second X to have that as an option. And so if we look at a pedigree, or sorry, a Punnett square, pedigree is on the next slide. If we look at a Punnett square for individuals on a sex-linked disorder, if we say that individuals who are color normal, both parents are color normal, the father isn't colorblind, the mother isn't colorblind, we have the chance of producing offspring. And there's a 50-50 chance for those offspring to be female or male, right? Inherit two X's or inherit an XY. And they're always going to get the X from mom. Doesn't matter if it's a daughter or a son. They get an X from mom and they get an X from dad. Well, if dad is color normal, he has the dominant allele because he is not having color blindness. He has color normal vision. And so all of his daughters are going to inherit his dominant allele for color normal vision. However, if they end up having a son who is colorblind, we know that mom carries the recessive allele. Mom is going to have a dominant allele and a recessive allele. We know she has a dominant because she's got color normal vision, and we know she has a recessive because she had a son who was colorblind. And the only way that can happen is if he inherited a recessive X from mom because he gets the Y from dad. So in this case, we absolutely can determine the sex-linked traits. There are some other sex-linked disorders. One that I think is really interesting is hemophilia. And so if you're not familiar, hemophilia is a blood clotting disorder, and it can be a bleeding disorder. When individuals have hemophilia, they are more often male than female. And some of the things suggest that hemophilia might be one of those traits that triggers a spontaneous abortion. And that can be because females who have hemophilia are unlikely to survive to adulthood, especially not when they hit puberty, because what is puberty? Well, for a female, it means you start bleeding every month. And if you have a clotting disorder, it means you have a hard time bleeding or stopping bleeding. You actually bleed more. And so you might bleed too much just by having your period as a female. Or if you go through childbirth is incredibly, incredibly bloody. And so obviously if a female makes it through that whole process, there's a chance that she will die in childbirth because stopping bleeding is hard in individuals who don't have a bleeding disorder in some cases. And so we're going to see that hemophilia is not very common in females. Males that do have hemophilia often don't historically make it to adulthood because, you know, anytime you get a cut, right, a bruise, you fall down the stairs, there's a chance that you might lose too much blood, especially if it's a bad sort of bleeding disorder. Think about the royal family, right? The Russian royal family. You might see something like that, okay? So that's something that we can see. I did happen to know a girl in college who was a hemophiliac and colorblind. which means that she is an individual who inherited two recessive alleles on that X chromosome. Now, this particular pedigree is a little interesting here. It is showing us that once again, this can be a recessive disorder, especially because it's skipping generations. Ignore this generation here for a second. We're going to ignore that this second son, the son that this first couple had, we're going to ignore his family lineage here for a second. So if we look at our first generation, the initial male is colorblind. Okay, he had a color normal daughter and a color normal vision son, and then the color normal vision daughter married the color normal vision son. But because her father was colorblind, we know that she is a carrier for color blindness, and so we do see a grandson down the road that is colorblind, and that makes sense because he could inherit that recessive X from mom. This male that is color normal from that generation did marry a female who was colorblind, and so their son was obviously going to be colorblind. There is a chance that their daughters wouldn't, but all of their sons would be colorblind. And, you know, it can happen, and especially with today's modern day medicine, if this was hemophilia, it is something that could happen. The girl that I happen to know had been on birth control since she was 8, in a way to prevent her from ever having a period, because they were very concerned what would happen if she did have her period. She also was colorblind, which was just an interesting thing that she had to deal with. She, if she ever got bruises, they were like, she bumped a table and they would be just the most giant bruises, and it's because her body had a really hard time stopping the bleeding from happening. Okay. Other X-linked recessive disorders besides color blindness are things like muscular dystrophy, hemophilia, not overly prevalent because, again, most individuals do have the chance of inheriting that dominant allele from mom. And yeah, check your understanding on these weird things with genetics. Again, this is not everything. Genetics is a much larger field of study than we have time to get into in Bio 110, but I hope that you found some of this interesting. And if you're curious, please go dig more. I strongly recommend going and learning anything you can. I think it's absolutely a fascinating topic. And if you have questions, please let me know. Thank you.